Today we will be discussing three main topics. First, the fusion of chromosome pairs at the start of meiosis, which is known as synapsis. Second, chromosome aberrations, which occur when a chromosome is missing, has extra or has irregular portions of chromosome DNA. Lastly, we will talk about the percent fitness between individuals based on their produced gametes. For this example, our individual has these two chromosomes. One of the first things you may notice is that these chromosomes are not identical, which means we have a chromosome aberration. By taking a closer look at these two chromosomes, we can see that the differing segment in chromosome 2 was removed, flipped 180 degrees, and inserted back into the chromosome. This type of aberration is known as a pericentric inversion because the segment that was inverted includes the chromosome centromere. Before I showed the synapsis, I wanted to point out that when there is a pericentric inversion, the normal chromosome must loop out. Unlike other aberrations, inversions will have an inversion loop formed from their second chromosome so that the sequences of the chromosomes will still align. Here we have the synapsis of individual 1's chromosomes. We can see that all the regions line up thanks to the loop of the normal chromosome and inversion loop of the second chromosome. For this example, there is crossover between the J and K regions to determine the 4 possible gametes that will form. I have labeled the chromatids in the example 1 through 4 to show the final gametes they will align with. For gametes, we see that number 1 is the normal non-recombinant gamete and number 2 is the non-recombinant gamete that includes the inversion. Both of these gametes are viable because they include all regions and the centromere. Number 3 and 4 are not viable recombinant gametes because genes are either missing or have more than one copy. Out of 4 gametes, since 2 were viable and 2 were non-viable, individual 1 has a 50% fitness. For our next example, our individual has these 2 chromosomes. You might immediately notice that these chromosomes are identical, which means we do not have a chromosome aberration. Even though there is no aberration, we can still proceed with synapsis. Here's the synapsis of individual 2's chromosomes. Since there's no difference between the chromosomes, the regions all line up with no loops necessary. For this example, there is crossover between the J and K regions to determine the 4 possible gametes that will form. I have labeled the chromatids the same way I did in the first example. With our gametes, we see that all 4 are identical. This was expected since there were no aberrations in chromosomes 1 and 2. The crossing over between the J and K regions occurred but provided the exact same gametes that the parent chromosomes had. All 4 gametes are viable since all regions and centromeres are represented. Since all 4 are viable, individual 2 has 100% fitness. Fitness is the ability to survive and reproduce. For both individuals, we were able to determine expected fitness. Individual 1 only has 50% fitness, meaning there's only half a chance of survival. Individual 2 has 100% fitness, meaning that all will survive.